Welcome to Performance Around the Clock, where we explore the latest research on how to keep our body and mind at our peak physical, emotional, and intellectual performance around the clock. I am your host, Professor Satchin Panda at the Salk Institute, La Jolla, California. On this podcast, we'll talk to leading scientists, clinicians, coaches, and policymakers about human performance. We'll cover different topics, including how different organs and different parts of the brain contribute to human performance. And we'll also learn how our lifestyle, that is, what, when, and how much we eat, sleep, and move around every single day, affect our health and wellness. I hope you'll join me in this journey to explore new knowledge and adapt it in our own life to improve our health and wellness throughout our life. Thank you, Hiro, for
Thank
joining
you.
us
Yeah.
today. So, for those of you who don't know Hiro, Professor Hiroki Ueda is at the University of Tokyo Medical School and also at the Riken, the premier research institute in Japan. Hiroki has been working on circadian rhythm and many other technology. And in fact, in science, we say technology opens and expands our imagination. So, in a way, Hiroki's work is expanding the imagination, what is possible in science. At the same time, he's also pushing the envelope. He's truly at the frontier of circadian rhythm research. Thank you, Hiroki, for joining us.
Yeah, thank you, Satchin. Yeah,
Sure.
it is my pleasure.
Yeah, so there are very few MD PhDs who work in this area of circadian rhythm.
Oh, I see. Yeah.
Typically, as MDs gravitate towards patient care or MD PhDs gravitate towards many other aspects of biomedical research. I'm just curious, how did you get attracted to circadian rhythm? And tell us like how it excites you.
Yeah, so when I was a high school student, I would say, get interested in say, physics and chemistry, as well as mathematics. Okay, and then when I start learning about the biology and medicine in Faculty of Medicine, University of Tokyo, I was so surprised. In biology, definition of time or definition of space is not well studied. So therefore, I thought, maybe, or we can start with maybe definition of time or space in biology, and then move forward to further complex things. Okay, I thought that would be kind of a very fast step, but I get interested in the biological timing so much, and then it's eventually become my kind of a lifetime, research topic.
Yeah.
But yeah, that's how I got interested in the biological timing.
Yeah. So, the term circadian rhythm can mean many things to many people.
Yeah.
Some people can say, when I tell them circadian rhythm, they immediately say, oh, you work on sleep.
Okay.
So, how do you communicate what is circadian rhythm to
okay.
your colleagues or maybe students to everyone?
Yeah, okay. So circadian rhythm is a kind of a, I would say, kind of a, or our simulation or how to say mimicking of the Earth. Okay,
Yes.
so we are living in Earth, and then interestingly, in our body we also have Earth, and then it's rotate and also revolves, and then our external environment is dynamically changing. Okay,
Yeah.
but it's predictable. So therefore, in our body, we have a day and night, 24-hour period of oscillation, and also we have a season in our bodies. So to mimic such kind of a dynamically and also predictable or say environment, we have a internal system. That's just 7 reasons.
Yeah. So one thing that you mentioned is predicted. So
Yeah.
that means a body keeps track of time and also predicts when it's going to be morning,
Yes.
evening, noon. midnight.
Yes.
Yeah.
And also, it's interestingly, it's also not only predict the kind of a timing, but also how to say represent some kind of uncertainty. Okay. So, for example, like me in San Diego or from Japan, I my internal body time is a little bit mixed up, okay, right now. So, because currently my internal body have some kind of uncertainty, okay. So, so in that sense, we can how to say represent some kind of uncertainty in our body, in our internal clock. Yeah. So in that case, our internal body's total amplitude will be a little bit decreased. And then such kind of a low amplitude can be represented as a kind of a desynchronization of the individual internal single cell level clock. Yeah.
Yeah. So that means almost, as you said, every single cell
Yeah.
has its own clock.
Yes.
And then when you travel from Japan to
Yeah.
here, that's how many hours of time change? Maybe?
Actually, I was in Houston first and then come down to San Diego. So almost 14 to 12 hours.
Yeah.
Yes.
Yeah.
Yeah. So
So it so there are two aspects you said. One is the clock helps us to predict. So when you're traveling, your clock was predicting it's going to be morning in Tokyo.
Yeah.
Whereas you were stuck in first in Houston and then now in San Diego.
Yeah.
And there was this uncertainty, and your body is still trying to deal with that.
Yeah.
So you have been here for almost a week now.
Yes. Yes.
But still, 1 week is not enough.
Not enough, right? Yeah. Especially if you want to advance your body time, I think maximum capacity to change your body time is 1 hour per day. Especially if you want to advance. So, if there is almost 12-hour differences, you need 1 to 2 weeks. Yeah.
Oh, okay.
At least. Yeah.
So that means even when people, those who are not flying like you or us, but they are staying awake maybe for 3 to 4 hours one night to take care of someone or to finish a task, or for students who are coming for exam, they are staying awake. So those 3 to 4 hours that they are staying awake and staying against their body clock, that means for the next 3 to 4 days, they may be still struggling to catch up.
Right.
Because it takes 1 day to adapt to 1 hour of
1 hour,
change.
yes. So if you wanna advance, if you wanna delay your internal time, maybe 2 hours would be a maximum. But I think there are 2 times differences. If you wanna delay, it's 2 times easier. But if you wanna advance, it's 2 times harder.
I see.
Yeah.
So it's much easier to delay your sleep time than to advance our sleep time. And many of us feel it that way because it's so easy to stay awake an extra hour or two.
Yes.
But it's difficult to go to bed early, and then ultimately you toss and turn, you cannot go back to sleep.
Yeah, of course, it's totally depend on the individual.
Yeah.
So I'm talking about the average. But if you go, how to say, work trip, I strongly recommend you to go west, how to say, west round.
Yeah. That's what we used.
Yeah.
Okay. Yeah, so you have a long career in circadian rhythm, 20, almost 20 years.
Right. Time fly by.
You and I kind of started around the same time, although. So your initial work was on what we call genomics.
Right.
That's what time of the day which genes turn on and off.
Right. Actually, my first work on circadian clock is modeling. Modeling of the cellular circadian clock.
I see.
So there are a lot of different genes within the cells, and then they are interacting with each other to make how to say some kind of a feedback loop, negative feedback loops. And then, but at that time, that was a model, and then I like to how to say measure and also test prediction by experiment.
I see.
Yeah, that is why I jump into the how to say fry how to say genomics first, and then move to the mammalian work.
Yeah. So, can you take us back and tell us what came out of those initial days of genomics?
I see.
Because at that time, if we dial back to 20 years ago,
Yeah.
We thought, or the community, the scientists were thinking maybe there are this handful of genes with whom you're modeling.
I see.
But then all of a sudden, the genomics, where we could measure thousands of genes at different time of the day, and you can figure out which genes are turning on and off. How did that change the field, or how did that change the impact of circadian?
Right. Yeah. So we learn kind of a simplicity as well as the complexity.
Yeah.
So in terms of complexity, so as you said, almost all genes, like more than 50% of the entire genome are oscillating, and then in different time, and also, or it's how to say, or it's reflect morning, day, afternoon, and the evening, and also night. It's almost continuous. Okay, that we learned. And then, interestingly, that kind of genes are, some genes are, say, common, conserved among the different tissues, but most of the genes are tissue specific, liver specific, kidney specific, brain specific. Yeah, we learn a lot by using genomics, and then we learn the complexity. But simultaneously, we also learn the simplicity. Because some core mechanisms are conserved among the different organs, and also by analyzing the regulatory region of the genome, we found kind of a morning element and daytime element, and also nighttime element, and sometimes these three elements are combined with each other to make how to say new timing, like evening, or maybe early day, or early night. Yeah,
I see.
Yeah.
So basically, what you and others found was it's not that genes just turn on in the evening or turn off in the morning. At any given time of the day, there are some genes who are waking up from their sleep, we can say that way, and then some genes are going to sleep.
Right.
And then the question is, if there is only one clock, how that clock is telling different genes when to wake up and go to sleep. So what you are explaining is you found there are different mechanisms, 3 or 4 different mechanisms. Each one codes for genes turning on in the morning, genes turning on in the evening or in the midnight. But what was more exciting from those early experiments that you nicely demonstrated, that you can even combine these switches, morning and evening switch, for example, and come up with a switch that will turn on genes at, say, 10 o'clock in the morning or 2 o'clock in the afternoon. And that's something that was never thought of, right, at that time. So in a way, you're going back to the same idea that pulled you in. What is the biology of time? Right. How time is encoded, how certain genes are deciding when to turn on and off. So that was pretty exciting. And then at the same time, you mentioned that different genes turn on in a tissue-specific manner. So that means some genes cycle only in liver, may not be in the kidney, or may not be in the brain. Right. And that's also an interesting aspect, because until then, people thought that if there is one gene that is doing a function in one cell, then it should be doing the function in every cell at the same time. Similar. But this work essentially told us that the gene may be part of the telling the time to the cell in kidney,
Yep.
but not telling time to liver.
Right.
Yeah.
So and also interestingly, that kind of a perspective was created not solely by a single researcher, but by the kind of a competition, community, and that was a competition and also cooperation, right?
Yeah.
So we contribute partially to create the kind of a larger view. That was a kind of an interesting time for circadian communities, and also interesting time for the kind of entire biology, because in terms of the timing aspect, circadian community kind of a little bit advanced. So because we have we use genomics tool to understand the kind of a dynamics. Not the static one, but the dynamic one. And also we try to extract some kind of simple or how to say mechanism out of the kind of a very complicated data. And also we have a go back and forth between imaging and genomics, or kind of a dynamical, how to say, assay and and genomics. So there are a lot of interesting research example were kind of invented at the time.
Yeah.
So we enjoyed basically.
No, I guess it brought another interesting issue opened up. That is, the genes that we're saying that are turning on in liver or kidney, they do some function.
Right.
And sometimes those genes turned out to be the genes that break down cholesterol.
Right.
Or make cholesterol.
Yeah.
For example, a lot of people are interested in mTOR pathway or autophagy pathway that breaks down stuff. And then there are also genes that encode different drug targets.
Yes.
So I guess the what started 20 years ago with all the genomics experiments essentially are now telling us that any drug target may be turning on at some time. And similarly, a lot of people are taking supplements, NAD supplement, for example, which is quite popular nowadays. But then circadian biologists, like as you said, many different labs doing these experiments independently, we're also validating each other's result.
Yes.
So you found something, and then maybe across the pond we found similar stuff, and then that built up that validation.
Yes.
And now, I guess 20 years fast forward, now it's almost accepted within at least circadian community. That almost every organ function peaks at certain time. Your medication should be at certain time. The supplement should be at certain time.
Yeah. And then, so by looking at the, how to say, such kind of a gene expression or maybe protein expression, so we try to, how to say, by using that kind of information, we can also measure the biological time by using the single time point assay. Right? At that time, or in the field, we have a kind of a classical method to detect the biological time in our bodies. So that is using the concentration of the melatonin. So data quantification of the concentration of the melatonin. And then that requires multiple sampling of the sample. And then if you track the kind of shape of the melatonin oscillation, you can tell what the sample's time is by observing the data backward. Okay, but if there is morning genes and evening genes or night genes or afternoon genes, one time point sampling tell you basically what the sample time is. So I kind of how to say come up with such kind of ideas and then apply to the mice liver or other organs first, and then expanded this idea to human blood metabolome.
Yeah.
And then many other people also apply this kind of idea to not only rodent but also to the humans.
Yeah.
Yeah. And then John Hogenish developed a very clever method to detect the human sampling time by just looking at the gene expression. So yeah. So and then now we know that a lot of human sample a human organ oscillate in vivo. So and then so as you said, many drug target are oscillating in humans. Usually this is very difficult to sample, but by observing the gene expression itself, and then which can tell you the timing of the how to say sample. So then you can speculate or infer almost all drug target are oscillating. Yes.
No, I guess this has a very immediate or very tangible medical or health benefit. So, for example, a lot of people they ask me, is there a test to figure out how is my body clock doing? And as you said, for to determine that, we have to sample melatonin, which is very tedious because in every 1 or 2 hours, you have to sample saliva or blood. It's also expensive asset.
Yeah.
And there is anyone just cannot go and get a prescription from a doctor to say, even if somebody is ready, even if they have thousands of dollars, it's not easy to get a prescription. Almost impossible. I don't think there is any medical test for consumer grade.
Right.
So this has been still a challenge to figure out from, say, our blood. If you can take just one time point and figure out how the clock is doing, because it's a very mathematical and biological question. For example, if some blood element, say A, reaches its peak in the morning, B reaches its peak in the at noon, and C peaks in the evening, then can you take one time point and figure out the levels of A, B, and C to see what time your body is telling you?
Yeah.
It's kind of like that, right?
Yeah, right.
And this is also interesting because A, B, and C may be coming from different organs.
Yeah.
So by just taking one time point, not only you can say how the body clock is running, you can also kind of deduce whether these clocks in liver, kidney, and heart are in right synchrony. Right. Just like a gearbox, are they in this right alignment or in misalignment? Right.
Yeah. So you're right. So I think because in our body there are different organs, and then each organ has an individual clock. So therefore, it's a little bit difficult to accurately dictate what's going on in our body. But basically, if our body is kind of in synchrony, we can tell what time your body is by taking that one sample. Maybe from the blood metabolite or maybe blood proteins. Yeah.
So how close are we to that point in time? Because it has a lot of applications. For example, if someone is an airline pilot, for example, before that pilot goes into the cockpit, if we can take one drop of blood and figure out, so you should be piloting this aeroplane for 2 hours, and then your body clock is telling you should actually sleep for 6 hours and then wake up. And the applications can be limitless. So how close are you to that point?
Yeah. So 2004, when I come up with such kind of an idea for the first time, I thought that would be immediately we can apply to the clinics. And then we find a pattern. Actually, at that time, I was working for the pharmaceutical company in Japan, Yamanouchi. Now become Astellas. And then we find a pattern. But after almost 20 years, it's almost expired. I see. So therefore, yeah, maybe my prediction will be wrong. But
No, I guess there is
we're
still
getting,
interest, right?
yeah, we are
Because
getting closer and closer.
we saw a couple of publications from Germany
Yes.
trying
Yes.
similar stuff.
Yes,
So,
Achim Kramer nicely, developed a good method, great method to take, how to say, measure the actually, protein level and also messenger level, and then try to tell the, how to say, internal time by using just one blood sample.
yeah.
Yeah, so I think, and also in community, after, I don't remember exactly, but a couple of years ago, there was a SRBR presidential symposium focusing on the single time point measurement of the internal clock.
Yeah.
So I was so happy actually, although I cannot get any, how to say, interest from the my patent.
Yeah.
But it's grown. Yeah.
No, sometimes you put a seed and then maybe other people will pick this up. And at the end of the day, it gives you satisfaction that
Right.
you started this.
Yeah.
That's exciting.
Mm.
I think let's just switch gear, and then you have done a lot of stuff in genetics of circadian rhythm or sleep.
Yeah.
And since our sleep, circadian rhythm, everything is encoded in genes.
Yep.
By mutating genes, we learn a lot. And also, as every human has few hundred mutations, we are also curious. As we know about our mutation, we'll also kind of guess what we may be susceptible to.
Okay.
So you have done some really remarkable new genetic tools to ask these kind of questions more rapidly. Can you tell us a little bit about this?
Yeah, okay. So around 2010, so I gradually start interested in the kind of a biological timing of the organism or brain. Okay, so therefore I'm get interested in the sleep-wake cycle, and then but to analyze organism biological timing is quite how to say labor intensive, and also it takes a time. Okay, so if you want to make a genome engineered mice to test your hypothesis, usually it takes 1 to 2 years because once you have an idea, you have to have genome engineered mice. But to make such kind of mice, you have to cross the mice a couple of times, like 5 to 6 times, and then finally you can get the final genome engineered mice. So I like to avoid such kind of a time limiting step, and then thought, oh, what if I can avoid such kind of a crossing or breeding? And then eventually to make such kind of mice within the single generation. And then we conceptualized such kind of a new technique as a next generation genetics.
Yeah.
At that time, in 2010, that was a pure concept.
Yeah.
And then it takes 6 to 7 years. And in 2016 and 2017, we come up with a new technique to make a knockout mice directly within single generation, or knocking mice within single generation. Yeah. So, and then by using such kind of technique, we try to test our hypothesis. Actually, every week. Yeah. And then 2016, we find out some kinase called CAM kinase 2 has a significant role in promoting sleep, non-REM sleep. And also 2018 to 2019, we also find out some acetylcholine receptor are really essential for REM sleep. So if you knocked out two muscarinic acetylcholine receptor, REM sleep are completely gone in the mice. Yeah. So by using such kind of a new technology, we find out that some important mechanism of sleep, non-REM sleep and REM sleep, can be discovered. Yeah.
That's it.
Yeah.
Yeah. So for most of us, I think the sleep field has gone through a lot of transformation in the last
Right,
20 years
right,
because a lot of people thought that sleep is when our brain is just inactive, and now we
Okay.
are learning that brain does
Right.
a lot of stuff during sleep.
Yeah.
And it's also becoming clear that the quality, quantity, and timing, all three aspects of sleep are important.
Right.
But when we go back to, these days, we always ask ourselves, when we cannot sleep enough, then we are asking, do I have a bad gene to sleep? If we wake up too many times, is my gene defective? or if I have weird dream, people also tend to think that there might be bad genes that make them see bad dreams. So since you are at the frontier of the sleep genetics, what do you think? How many genes that we know now that affect quality, quantity, and timing of
I
sleep?
see. I see. Yeah.
Is it a
So
dozen?
more than that, I
More than
suspect. Yeah, probably. So it's already couple human-related sleep genes are discovered, but basically, how to say, contribution is not so large. When we compared with the uh, genes, sleep-related genes in other organisms like mice. Okay, so we in circadian clock, in human genetics and also mice genetics are overlapping. Yeah, for example, period genes or other cries also discovered in both organism, but in human we still some genes are discovered in human, and then couple genes are discovered in mice, but it's not overlapping with each other. So therefore, I think still there are a huge gap between two organs at least, and then I think huge gap in understanding of what kind of a genetic aspect of our sleep wake cycle. So,
but I guess
yeah.
it's also very challenging because in humans, for example, nowadays, there are many things that affect our sleep that are genes.
Right.
So when people cannot sleep at night, it can be anything from how many cups of coffee or tea they had. And sometimes people say they don't drink coffee, but then they may be consuming stuff that has caffeine. So, for example, Diet Coke.
I see.
A lot of people don't know that there is caffeine there.
Okay.
And similarly, dark chocolate. I don't know about matcha. It also has
matcha has a caffeine, yes.
Caffeine. But for a long time, people think that, okay, if it doesn't look like coffee color, then so that's one. Second is there is also a lot of light people are exposed to. So from the human side, there is now challenge to really filter out all the noise of coffee and other things to see what is the contribution of this. Right. So, for example, a lot of people do have good genes, but when we eat unhealthy food, we all can gain weight. So even the good effect of good genes can be blunted by bad food. So similarly, even if we have some good genes for sleep or bad genes of sleep, it becomes very confusing to understand the contribution. On the other side, I guess that's where you are leading is to see whether mice, if we can use laboratory animals, can we understand their genetics? But then mice don't sleep like humans.
Yes.
And it's also very challenging to monitor when the mouse is sleeping or when the mouse is dreaming. So is there a new technology to monitor sleep in mice?
Yes, actually, usually to work on mice, how to say, mammalian sleep, mice, rodent sleep, you have to do a kind of a brain surgery because mice don't like electrode. So so therefore they took off or they bite.
Yeah.
So therefore, we need to have some kind of a brain surgery onto the mice to accurately measure the mice sleep-wake cycle. But it's labor intensive and also it requires skills. Okay, so that is why we developed a new method to monitor the sleep-wake cycle by using the respiration of the mice. Okay, and then if the mice inhale or exhale, uh, surrounding air pressure will be slightly changed. So we detect such kind of a subtle change of the air pressure by using the piezo sensor, pressure sensor. And then non-invasively monitor the respiration patterns between sleep and wake, and then convert such kind of a data into the sleep status or wake status. So by using that, so now we can accurately monitor the mice sleep and wake.
Wow.
Yeah. And then in Osaka, Riken, we have 1,000 cases dedicated for the mice sleep-wake cycle. So by using that, we can now capture the at least my sleep-wake cycle.
Yeah.
Of course, of course, they are nocturnal animal, which is unlike us.
Yeah.
But we learn a lot.
Yeah. No, I guess this is very important for research because doing a surgery in mouse and putting those electrodes inside brain, it's not trivial. Very few people in the world, maybe few dozen people in the world, can actually do it successfully, so that the mouse is alive at the end of the surgery. And also these probes cost thousands of dollars because once we tried it, it's almost a couple of $1,000 to measure sleep from a mouse for a few days or weeks. So this is transforming science by enabling people to scientists to measure sleep. So that's one aspect, that just by looking at how the mouse is breathing, because when we sleep, our breath also slows down, right? In humans. So from there, you can figure out whether the mouse is sleeping or not. But you can also use the same technology, for example, to figure out whether the mouse has sleep apnea, right? Because the mouse may be snoring, cannot breathe. And
I see.
Can you or no?
I'm not. I have not yet think about that, but maybe meditation or maybe hibernation. Yeah, or respiration itself. Yeah. So by using similar technology, same technology, you can detect the patterns, different patterns, and then tell you or investigate the biology or mechanism of such kind of a phenomenon. Yes.
Yeah, but you brought up this interesting point that in mouse we might find genes which contribute to sleep, but in humans their effect may be very small. Or maybe is it possible that some of the genes that we find in mice that contribute to sleep in humans, if they're mutated, they may cause other type of disease? So, for example, we know that there are some birth defects. People are born with birth defects. Unfortunately, these kids um, have other brain issues. But in many of those cases, they also cannot sleep well. In many cases, they are sleeping at the wrong time or sleeping inverted. So, sleeping during daytime, awake during night, or they have long sleep or short sleep. So, is it possible? But the severity of growth and development or speech, all of these are so severe that the physicians don't pay attention to how their sleep is.
I see.
So is it possible that by looking at these extreme cases where these kids who have more severe symptoms?
I see. Yeah, so there are a couple of such kind of diseases. Not only developmental diseases, but also like autism, neurodevelopmental diseases. Most of the case have a sleep symptom, and also in other diseases like psychiatric disease or neurodevelopmental disease has also all sleep-related symptom. So therefore, I think you know sleep can be used as a kind of a high sensitive sensor for your brain status because your brain activity is so high. Usually, it's become uh, awake, and then so low, or it's become longer sleep. So therefore, uh, how to say, sleep-wake cycle monitoring can be used as a kind of a high sensitive sensor for the brain activity. So that is why we try to develop the new method for
I guess
that.
let's focus on this one. What you were saying is very profound.
Mm.
What you're essentially saying is almost every human condition
Mhm.
where a brain is affected,
Mm.
whether it's autism spectrum is one, ADHD is the other
yeah,
one,
yeah,
and of course all the developmental disorders
Yeah.
that people have
Yeah.
difficulty speech and language development, but also depression,
yes,
anxiety,
Yeah.
and even people who get severe migraine pain. Um,
Pain
so
also,
it
yes,
almost sounds like if there is something wrong with our brain,
brain, yeah,
that should show up
Mhm.
in something wrong with our sleep.
yes,
Right.
yeah,
If there's something wrong with our brain,
Mhm.
then something goes wrong with our sleep.
Yeah.
Wow, that's that's a very big statement, and I think as you were talking, I was trying to remember, I was trying to figure out: is there any human brain affected disorder which does not involve
In
sleep disorder?
I cannot find any. So
Yeah.
therefore, or I think, in daily life,
Yeah.
if you can monitor sleep-wake cycle in very accurate manner,
Yeah.
which is usually difficult, but if you can do that, maybe that kind of data tell you or tell the doctor about your status of brain.
But I guess
yeah.
you emphasize this point that accurate, because nowadays there are many wearables. Even my phone sometimes tells me how I slept, but it's all based on whether I am moving my phone or not, whether my wrist is
Right.
moving or not. Uh, can you tell a little bit about how this current technology that's in many devices, even I'm now wearing something, it tells me that I didn't sleep well. But anyways,
Okay.
the point is, what is the technology? When we see it's telling us how we slept,
Okay.
what it is exactly telling,
Okay. Yeah.
and let's dial back and say quality of sleep and duration of sleep, because we know go to bed and maybe waking up from beginning to end, that's the duration. But then we always hear this, whether you're into REM sleep, non-REM sleep, stage one sleep, stage two sleep. Let's dissect that.
Okay. Yeah. Okay. So
All of us.
okay. So let me take one example, like a two-stage judgment, just sleep and wake. Okay. Because uh, kind of a wake after you go to bed, tell you about the quality of sleep a little bit. So therefore, that would be a good example. Okay. So current technology like Fitbit, Apple Watch, or Oura Ring, or other or acceleration-based method is not good at accurately detecting the awake after you go to bed. So usually sensitivity, which means that accuracy to detect real sleep is usually good. But specificity, which means accuracy to detect true wake, is not so good. Okay, so usually 50% of the time, uh, your current technology have a wrong answer. So therefore, it's not good at how to say detecting the how to say quality of sleep by using current well. So that is why we decided to develop the new method, new algorithm, uh, to detect sleep and wake in very accurate manner by
Mhm.
using wearable. Yeah.
I see.
And then around 2022, actually this
This
year,
year.
yeah, early this year, we published a one paper to propose almost more than 90% in sensitivity and then 80% of the specificity can be achieved by just looking at the movement of the your arm during wake, during sleep.
Mhm.
Yeah.
So that means you can actually take data from even current wearable,
Right.
Fitbit or a Ring,
Yeah.
and your algorithm will be much more accurate
Yeah.
in wake and sleep.
Yes. And then which tell you about the not only duration of sleep, but also or amount of sleep, but also quality of your sleep. Yeah.
Quality.
Yeah. And then after that, we also apply this technology not only to couple individual, but also to the 100,000 of the individual, which is recorded in the United Kingdom.
Mhm.
For the 40 years old and 50 years old and 60 years old.
Yeah.
And then we classified individual life in terms of the sleep-wake cycle. And then we found some people have a very long sleep, some
Yeah.
people have very short sleep, and some people have shift work
Yeah.
type, and some people have kind of a non-24 hours. They are don't tracking the rotation hours. And also some people have severe insomnia, and then some people have kind of a neurodegenerative disorder like sleep. So based on that kind of a I would say finding, we at that time we also proposed the annual health checkup, annual sleep checkup.
Yeah.
Because that kind of a data tell you about that kind of a brain status.
Yeah. So this set of data that you're talking about, this is from UK Biobank, right? So
Right.
this is UK Biobank, which is a large study, and there are 100,000 people's active watch data,
Yes.
actimetry data.
Yeah. Observation data. Yes.
And that must be like more than 1 million days of data.
Mm-hmm. Yeah.
That's a huge, I think that's the largest such data set that
Yes.
anyone has computed.
Right.
So, other than 10 or 20 or 30 different types of people based on their sleep,
Actually,
what is the big
16. We classified into the 16 different type of the.
wow,
Yeah.
16. It's not early bird and late night owl, but actually there are 16 shades
Yeah.
in between.
Yeah.
Okay.
So, yeah. So, and then, so that is why, so we thought, oh, now we have a nice resolutions to classify the people, actually healthy individuals.
I see.
And then, so that is why, such just two-stage accurate sleep-wake cycle measurement, sleep-wake stage measurement is actually good enough to have such kind of resolution. So in Japan and also in some other country have annual health checkup.
Yeah.
So in Japan, because I mean living in Japan, so we propose the annual health checkup because it's how to say feasible and also wearable is not so how to say expensive. So therefore we can measure at least 1 week in a year and then try to check your brain. Yeah. Usually, such kind of a measurement can tell you about what's going on in your brain in terms of the health and disease. Yeah.
Yeah. So you said that sleep is the marker or the phenotype. Sleep is the window to our brain almost. So if there is something wrong with our sleep, then it's a red herring that there is something abnormal going on with our brain.
Yeah.
And when something abnormal going on with the brain, it's most likely that since brain controls a lot of our organs, then something may also be going wrong with our body.
Yeah.
Having said that, and since you're an MD, what is your definition of good sleep
Yeah.
for an adult?
Okay. Okay.
Like a 50-year-old.
Okay.
I'm very selfish here because I just want to know.
I see. Short answer is we still don't know yet. But we in the process to define good sleep. Because that is kind of a one reason why we try to introduce annual health checkup. Annual or how to say sleep measurement into the health checkup. Yeah, because we can take a blood biochemistry simultaneously, and then because to define good sleep, you need to have data to define good, okay, or health.
Yeah.
And then we also have accurate. We also have to have accurate sleep measurement, which we can provide. Okay. But statistics or a previous study tell us about like usually 7.5 around 7.5 sleep is have a long kind of a longer how to say lifespan.
Yeah.
And then less than that or more than that you have a how to say shorter lifespan.
So it's almost like,
yeah, bell shaped. Yeah.
so if you have somewhere between 7 to 7.5 hours of sleep,
Yeah.
that means you are likely to stay healthy and live longer.
Yeah. So I don't know that is a consequence or just or correlation, but yeah, previous data suggests such kind of a
Yeah, so this is epidemiological data, but we don't know. For example, those who are sleeping 5 hours, if they switch to 7.5 hours sleep, we don't know whether that will improve their diabetes, heart disease, and cancer, and all this stuff that they're already suffering from, not reducing risk. But so that's the difference between correlation and causation.
Right. So yeah, right. So but now Japanese society of sleep get excited about that my proposal like annual health checkup, sleep checkup, and then if we how to say implement such kind of a annual sleep checkup together with the health checkup, I think we can measure the biochemistry, which indirectly tell you about that how good your sleep strategy is.
Yeah.
Yeah. Because we can monitor the individual let's say individuals biochemistry change over the time. Yeah.
But now, right now, it's there is no harm in telling that most healthy people, to maintain good health, they should try to sleep 7 to 7.5 hours.
Yeah.
Particularly those who are not sleeping enough. Is that as a MD, would you suggest to your patients that they should try?
Yeah.
7 to 7.5 hours?
Sleep is kind of a how to say one of the few uh, how can I say uh, uh, kind of item which if you fulfill your kind of a greed to have sleep, that's a good thing, right?
Yeah.
So for example, kind of taking a food is not so easy, right? So if you fulfill satisfy your greed,
Yeah.
you're gonna have a problem, right?
Yeah.
But for sleep, if you fulfill your needs or greed, I think it's a good thing. But
Yeah.
usually it's end up with 7.5 or maybe 8 hours or 7 hours.
Yeah.
It's depend on the individual.
Yeah, of course. It all depends.
Please follow your natural greed in terms of sleep.
But I guess what happens in modern life, we are tied to going to school,
Right.
and then to office, and then we have other responsibilities. So we rarely get a chance to fulfill our sleep grade.
Right.
So I guess the ideal experiment would be just go on vacation or unplug yourself from digital devices and take a break from your work, and then for 4 or 5 days, just try to sleep as much as possible,
Right.
and then see where do you fall
Yeah.
and what is your sleep duration. Because I think recently I talked to Dr. Horacio Iglesia, and Horacio told me this very interesting experiment that was done, that is to put teenagers in this isolation kind of relaxing environment. And what people found was teenagers initially they have sleep debt, so they'll sleep for 10, 11 hours, but after 4 or 5 days, they came to around 8.5 to 9 hours of sleep.
I see.
So maybe that's what we all have to do.
Okay.
Take a week off and figure out what is our ideal
Right.
sleep.
Yeah. So maybe it's like as you said, like in free environment to measure and then measure their sleep, or maybe just take how to say individual together with the health marker like blood biochemistry, and then to define the maybe ideal sleep. Yeah. So either way it works. Yeah.
Okay. Because there are some really interesting studies showing when people who typically sleep or they need 7 hours of sleep, when they're forced to sleep for 4 or 5 hours,
I see.
then they develop early sign of diabetes, right?
Yeah.
Okay,
right.
So in that way, for all of us who are going for our annual health checkup, maybe it's a good idea to get good night's sleep for at least a week to see what is our.
Yeah, so usually, during the weekday, you usually have some kind of a constraint, or I would say, you're you have what you have to. Yeah, I think another method to know what is so your sleep is how to say sufficient enough is just how to say compare that weekday sleep and also weekend sleep. Yeah, because usually you have a how to say during the weekend you have a task, or you have what you have to do, right? So but in the weekend you have a more relaxed environment. So if there is no change between weekend and weekday and weekend, I think you have a good amount of sleep even in the weekday. So just compare the weekday and the weekend sleep habit. Yeah.
Okay. Now, these are very useful tips.
Yeah.
So now we talked about the number of hours, but there is also this idea of quality of sleep, and people talk about slow wave sleep, deep sleep, and REM sleep. Is there a good approach now to figure out how much deep sleep or REM sleep we are getting? Because different devices give me different readings. So what is the gold standard now, or should we not worry about that and first just go for the number of hours?
Yeah. So currently, usually, or consumer devices is not good enough to detect sleep and wake. And then I cannot expect, they already achieved the high quality for the deep sleep and light sleep. So usually to define the, how to say, such kind of a sleep stages, you need an EEG and EMG. There is a home devices. If you use such kind of a devices, I think you can tell, but the current consumer device is not good enough. I hope we are in we can how to say come up with the new method, not only to detect sleep and wake, but also detect sleep, non-REM sleep, REM and wake, or deep sleep, light sleep, and REM sleep and wake. So 3-stage detection, accurate detection, or 4-stage accurate detections. Yeah.
But is there just like we kind of discussed that 7 to 7.5 hours of sleep seems to correlate very well with health. Similarly, are there studies that have come to any conclusion about how many minutes or hours of deep sleep?
I think it's very difficult because usually EG and EMG can be measured only for the how to say one night, right? And also mainly measured for patient, not the healthy people. Of course, there is a study, but it's not 1,000 or 10,000 scale. So therefore, I think it's not good for statistical power. So yeah, but recently we tried to how to say implement annual health checkup for wearable, and also how to say kind of a brain doc type project, in which you can measure the EG and EMG for the healthy people together with the blood test and the wearable. So if we successfully implement such kind of how to say annual sleep checkup in Japan, maybe we can tell the good statistics. Yeah.
But I guess this is a place where technology hasn't changed much in the last 20 years to make sleep measurement, accurate sleep measurement in terms of deep sleep or REM sleep, scalable, right? I mean, so we still have to depend on somebody coming into a sleep clinic and sleeping there overnight with bunch of electrodes.
Right. So, but there are some update. So nowadays, you don't. Of course, it's depend on the researcher, but very small amount of electrode are needed to accurately detect the sleep and wake, and also non-REM and wake or full stage of sleep wake cycle. So you only need a very small number of
one electrode.
one was and then therefore home PSG are now possible. Okay, of course if you're hardcore sleep researcher, you think that's not good enough.
But still, the home PSG can extract my deep sleep or REM sleep and how many hours or minutes I'm in state.
Or at least we have a hope toward that kind of a goal. Yeah, that is why I feel there's some kind of a good potential for home PSG.
I see.
And then if you can collect the data in home PSG, you can improve the wearable accuracy. So
PSG stands for polysomnography.
Yes. So usually multiple electrode PSG, like more than eight electrode PSG or EG, EMG, tell you about the how to say is a kind of a gold standard in this field, and then which can be surrogated by the 3 electrode or one couple of electrode EG and EMG, and then which can be surrogated by the wearable. So
that means when our wearable tells that we are in REM, that's essentially we're moving our hand.
Right. Or
breathing or blood
actually, yeah. So pulse is kind of a frequency a little bit changed during the REM sleep because our sympathetic nerve or activity are slightly changed between REM sleep and non-REM sleep. So therefore, your pulse information can be used to how to say detect REM sleep and non-REM sleep. Yeah.
But why do we need so many electrodes attached to our skull to figure out whether our brain is sleeping? Means can we just connect one? And why do we need so much electrodes? And then second thing is when we are collecting this information, how frequently we are collecting? Once a second, 100 times a second, or 1,000 times a second. And why that is necessary?
Usually, 100 hertz.
So 100 times a second.
Yeah.
So every second we are collecting 100 time points.
Yes.
100 measurements,
Yeah.
which is enormous. For example, if I want to go get my cholesterol done, it's one time in the day, and it's just one measurement. But for sleep, we have to measure 100 times a second, and now throughout the night, if I'm in bed for 8 hours or 9 hours, then it's just mind-boggling how many data points are there.
Right.
And this is only from one electrode. And then you're talking about multiple electrodes attached to the brain. Why do we need so much data?
I see. So maybe we can reduce the electrode down to couple of electrodes because it's many paper already come out. Because multiple electrode include at least one electrode. So therefore, by using the more than 8 electrode teacher data, more than 8 electrode data as a teacher data, and then try to predict that such kind of a teacher data by just using one electrode. That's possible by looking at the existing 8 electrode data. So, and then many people says, just one electrode would be good enough. Of course, you need some kind of a how to say threshold, but many people said one or few electrode would be good enough. So we have a hope to reduce electrode, and then frequency maybe of course we usually we take 100 hertz or more than 100 hertz for electrode measurement. But maybe we can also reduce that data, especially usually wearable data are collected like 25 hertz.
Okay. Yeah. 25 times a second.
Yeah. And then it's good enough for the tracking the pulse. Of course, it depends on the sensor, but I think, or in that sense, maybe you can also reduce the time point, especially for the wearable data. So, yeah.
No, maybe you can just like for mice, you have built these cages where mice sleep and wake up and breathing, you can figure out. Maybe you can build an apartment suite. We all will come and because this is also becoming a challenge. Many hospitals, many medical centers actually don't have sleep clinic or they don't have sleep measurement suite where people can go and sleep and measure. Or alternatively, if you come up with an maybe commercial product that can be sent to home, home PSG is made more affordable, and then that will transform this field. Then we'll actually learn how quality of sleep correlates with health. And when you're collecting all this information, it's also likely possible, not only during our sleep, in our wakeful time, it's likely possible to figure out whether there is something wrong with our brain. For example, early sign of Alzheimer's disease or Parkinson's disease or any other disease. Or maybe better diagnosis of ADHD, autism. All of this.
So currently, now wearable data can be measured every years or once in a year. Maybe if you're worried about your brain status, maybe it's a little bit expensive, but the home PSG would be possible. And then we are now trying to implement such kind of a health system in Japan. And then we'd like to extend this kind of activity not only in Japan but also in other countries. Maybe in US or maybe in Europe. And then so for that purpose, we need to improve the kind of a algorithm to accurately detect the sleep status by using the very few electrode or just using the data from your arm. But I think it's possible. And also it's beginning right now.
I mean, going back to what we started discussing, one time point blood biomarker to look at how the organs are doing and how they are synchronized, or the clocks are synchronized, and now with the home PSG, I think in future the challenge will be how to process this data.
Right.
Because how long does it take to process, say, one night or 7 nights of PSG data?
Yeah, okay. So time limiting step is not the computation-based analysis, but currently, most limiting step is a human annotation, human-based annotation. Of course, it can be probably in the future, probably that would be almost fully automated. Of course, currently, qualified kind of a technician, yeah, can only annotate. Yeah, that's allowed. And then also responsible for the kind of a quality of the data. But once such kind of enough data accumulated, maybe we can fully automate it and then at least support for their annotation. So nowadays they can annotate the couple one night data for a couple hours. But maybe we can increase the throughput of the annotations. Yeah.
So we're talking about looking into the human brain from outside.
Yeah.
But you are also at the forefront of this new line of research to look inside the brain. Of course, not in humans, but in animals.
Yeah.
Because if we think about major advances in medicine diagnosis, they all relate to looking inside our body. For example, starting from X-ray. X-ray allowed us to see whether the bone is fractured or it's a minor fracture or what happened. And then you fast forward, we have CT scan, MRI scan, all of these scans. But even functional MRI, but they're not letting us look into one cell at a time. So tell us, what are the challenges and what you have been doing, even if we cannot do it in humans?
Okay.
In many cases, animal research helps us to figure out. So
yeah, okay. So let me start with why we start the whole brain imaging. Okay, so 2010, as I said, we started the sleep research. But at that time, unlike circadian clock, in which we know that there is a center of the circadian clock located in the suprachiasmatic nucleus, in sleep research, nobody knows what is a center. It is localized in a particular center or distributed among the cortex or other brain region. So therefore, we have to do some kind of unbiased study. So that is why we decided to develop the new method to look at every single cell existing in the mice brain, at least. Okay, and then it took 4 years, but we tried to clear the brain. I mean, in other words, to make the entire brain become transparent, and then try to look at every single cell existing in the mice brain by using the kind of a customized microscopy called light sheet microscopy.
How many cells are in a mouse brain?
100 million cells. Yeah.
100 million cells.
Yeah, slightly less than current Japanese population, maybe future Japanese population. Yeah, and then in liver, and then interesting, such kind of technique can be used not only to mice brain but also other organ like a liver, in which we have more than 355 million cells. Yeah.
Oh.
Yeah. Which is
so the liver is bigger than the mouse brain. Mouse liver is bigger than the mouse brain.
Yeah, and also that number is slightly larger than I think entire USA population. Yeah. So yeah. But
so what have we learned, or what are the promise of learning more about the brain from this kind of imaging, which is very cutting edge, because typically we look at few hundred or maybe few thousand cells, and you're looking at all 100 million cells.
Yes.
And not only you're just looking at cells, you're also looking at which genes may be on, which genes may be responding to, say, sound, light, stress.
Right.
Right.
So, for example, when you, how to say, induce diseases in mice, like a schizophrenia, and then mice brain activity is hyperactivated. Okay. And then if you treat that mice by using the currently available antipsychotic drugs, and then you can suppress some of the activity, but there is a residual activity. Okay. And then in human, we can also treat some kind of a symptom of the schizophrenia, but there are still remaining symptom which is untreated yet, like a negative symptom or memory deficit. So now we and other researchers who using our technology try to look at what is remaining by using currently available drug, and then targeting on such kind of a remaining activity to develop new drug, because that's a kind of a that tell you that this is a real time. Yeah.
Yeah, this is very exciting because for many drugs, the saying goes there are old drugs with side effects and new drugs for which you don't know the side effects yet.
Right.
So by using this technology, you can create the disease in, say, in a mouse, and then treat the mouse with the drug, and then go back and look at which part of the brain is still abnormal.
Right.
And that's something that we cannot do in humans.
Right.
There's no way we can do that in humans. So this will really push the frontier of science, neuroscience, psychiatric disease, any kind of brain disease. So similarly, maybe, for example, one thing that comes to my mind is migraine pain. There are many different types of migraine pain, but we don't know where does it start from and where does it propagate.
Right. And also, like neurodegenerative diseases, there are mice model in which human mutation are introduced. Okay. And they usually that kind of a mice eventually have neurodegenerative disorders. But in at the very early stage of their diseases, we can see the defect. So, if you can target on the very early symptom observed in mice brain, maybe we can access to the mechanism, or we can access to the kind of a entry point of the treatment. For example, such mice in such mice, we can measure every single cell, every single neurons, and also every single immune cell in the brain, like a microglia. And then microglia are literally eating the neurons. So we can measure such kind of a phenomena in very accurate manner. So in
the whole brain.
Yeah. So actually, out of the 100 million cells, actually half of the cells are neurons, like a 47.2 million cells are neurons, and then 3 million cells are microglia, which is immune cells. And then such microglia are literally eating the neurons.
Yeah, because they are almost like they have tentacles, they go around and then can capture and eat them.
And then you can detect such kind of an unusual event in the entire mice brain. So it occurred in very sparse manners, especially in the early stage of the diseases. It occurs, but it's very sparse. So therefore, it's very difficult to detect by dissecting, sectioning. But if you can look at the entire brain, you can accurately detect such kind of event. And then also you can compare the drug-treated groups and the untreated groups. And if you can target on the very uh, beginning of the diseases, you have a good chance to treat the not only mice but the mice disease but also human diseases. Yeah.
So this also requires new type of microscope,
Right.
right? So are you building those microscopes?
Yes, actually, so we have a good physicist and the engineers to build. So therefore, if you wanna, how to say, develop the new technology, definitely you need to collaborate with the different researcher with different discipline, or you, you you have to learn the different discipline to integrate uh, two or more discipline in your brain. Yeah.
Wow. So you are an MD,
Yes.
PhD,
Yeah.
and you started with physics, chemistry, mathematical modeling, gene expression, and then developed all these fancy tools from mouse breathing, from where you're figuring out sleep, and then now looking deep into the brain with new kind of microscopy.
Yeah.
This is very unusual and very unique for a
But
career, right?
yeah, but medicine itself incorporated many disciplines because the goal is understanding and also controlling the how to say our healthier or health status, right? So goal is just always same, but method or tools are always different, over the time, So therefore, I just follow that kind of a medical trend or medical principle, right? So, because to understand or to treat, you have to use or you have to learn whatever you have to do.
Yeah,
right?
yeah.
So that's a very simple rule.
You are very humble,
Yeah, but I'm teaching on the pharmacology in my university, University of Tokyo Medical School, and then to teach pharmacology, you have to control the molecular property, and then to control the molecular property, you have to learn how atoms are creating the molecules, right? And then such molecule expressed in the particular cells. So therefore, you have to learn the cell biology and also kind of sometimes anatomy and physiology. So to control something, you need to learn not only atom but also molecules, cells, and then organ or even organism, right? So that's a kind of a beauty of the medicine. Sometimes I'm struck with the complexity of the such wide width of the larger width of the how to say discipline, but sometimes I uh, just surprise and enjoyed such kind of a interaction among the different disciplines. So
Yeah. So I guess you're right. Like human body is the most complex system that we have seen, and it's also most enduring. So, for example, almost everything that we see on this table, and even this cell phone, we appreciate this cell phone so much. But my hand that's holding this cell phone is older, much older, and self repairs itself, self recharge itself than the cell phone. So we still don't understand many aspects of human health and how we is, what determines our lifespan. And that brings us to this overarching theme or goal of all biomedical research to enable everybody to live in their peak performance, physical, mental, and emotional performance at any age, at in any gender, nationality, and it's almost like a human right and human aspiration.
Yes.
So along that line, we know that Japan enjoys one of the best longevity in the world. It has I'm not sure what they start recently, but it has the highest number of centenarians too. But at the same time,
Mhm.
there is a confounding data that Japanese sleep less than
Right.
many other countries.
Yes.
So if sleep is so important,
Uh-huh.
how come Japanese people live so long?
Okay. You're right. We are, notorious for bad sleep, right? Insufficient sleep. But I think we can live longer or healthier than current stages. Yeah, at least I believe so.
Yeah.
So, uh, yeah. So, we have a lot of problem, including sleep problem. And if we solve the problem, we can live longer and healthier and survive.
Yeah.
Yeah, yeah. So, yeah, to prove that kind of a hypothesis, I think we like to. I like to improve Japanese entire sleep,
Yeah.
and then compete with the USA. Yeah. Or
Yes.
Europe, yeah. Yeah.
Yeah, so that's always a conundrum. People always ask, how come? But is it possible that the centenarians, who are now centenarians, maybe they slept better in the early part of their life?
I see.
Maybe for the first 70 or 80 years, they were sleeping much better, and now the current Japanese adults who are sleeping less, are they going to sleep? Are they going to have a shorter, healthy lifespan? Um, so that's also an interesting point to consider to see whether
I see.
longevity is plateauing in Japan or is it still going to go up? And
I see. Yeah, so still slightly going up.
yeah, it's still
Yeah,
going up.
yeah.
The last time I checked.
Right. But and yet there is a correlation between the lifespan and also sleep status. So therefore, or maybe or we are getting such kind of data gradually because we are now implementing the annual health checkup, then try to look at the occurrence of the diseases over the time, and also hopefully in near future uh, lifespan data. I hope I before I get too old, I can get such kind of data. But
Come on, here you look the same 30 years ago. You don't age. But coming back to this aging, I mean, you are an MD, you are looking at human health, and you travel around the world, you are familiar with problems in many other countries. What do you think is contributing to longevity in Japan? Is it the food? Do you have good quality of food? Is it the amount of food? Are the portion size smaller and people are eating less? Is it physical activity? Because in Japan, not too many people actually drive. And, in the US, I walk from my kitchen to the garage. Whereas in other countries, I have to walk from the hotel room to the nearest bus station, train station. And you have all these activity tracker. Do Japanese people exercise more physically? They're more active? Are they eating less? Is the quality of diet much better? Is that less processed food and less
I see.
bad fat? What are the secrets?
But we are now getting worse and worse. Like food is getting westernized, maybe influenced by the fast food culture imported from maybe US. So we have to cope with such kind of trend. So yes, currently we are in good status, but I don't think that without any intervention, I think it's going to be worse and worse. So we have to cope with that, and also, how can I say, or Japan, also, many young people or uh, use getting less how to say exercise, getting less exercise. So therefore, I think or we need to have good measure to how to say improve the such kind of exercise as well as a sleep. Yeah. So, oh yeah. So and also because of the COVID-19, kind of a uh, it's it's changing. I mean, lifestyle is also changing. Oh, I don't know yet.
What specific aspect is changing?
Uh, because we don't know how to say commute, meaning maybe less exercise.
I see.
Yeah, of course it's good for your sleep,
Yeah.
but we are how to say not move a lot. Yeah, so I don't know that's gonna be in good. How to say it's a mix. Yeah, we have a longer sleep.
Yeah.
But I get exercise.
Less
and then
exercise.
that's exercise. Yeah, and then quality. I have no idea. We have not yet have a data on sleep quality. Yeah, because if you have a good exercise, you will have a good quality of the sleep. Yeah,
You sleep more deep.
Yeah.
Yeah.
So therefore, it's mixed trend. Yeah.
Yeah. So and then you are also currently the secretary for
I see.
the
Yep.
Society for Research on Biological Rhythm, and you have been playing a leading role in Japanese Sleep or Circadian Rhythm Society as well.
Vice president. Yes.
So this gives you a very different perspective because it's beyond your own research, it's beyond your own research team or university. You're looking at what are the challenges, health challenges, not only in Japan but all over the world, and how this small group of researchers, because if you look at circadian rhythm researchers, there are only maybe 500 of them in the entire world, which is smaller than the number of doctors in University of Tokyo or Keio hospital.
I see.
So it's a big responsibility.
Okay.
So how do you see
I see.
sleep and circadian rhythm researchers play a role in elevating or improving human health? Because, as you said, when sleep is disrupted, then so I guess the point is, how do you see that the small number of people who are doing research on sleep and circadian rhythm, and as we know, as we discussed, that if something is wrong with our brain, that shows up in our sleep. Similarly, when something is wrong with our sleep, that shows up in our body. So how circadian rhythm researchers, starting from students to people who are getting ready to retire, can contribute to elevate the health of the planet, global health by the research? Yeah, so I think circadian rhythm researcher and also sleep researcher may have a unique role because sleep are involved in the many actually a lot of psychiatric diseases and also neurodevelopmental or neurodegenerative diseases. So to understand sleep or use sleep, let's say knowledge, you can apply that kind of information to the many different aspects of the diseases. And also, circadian rhythm, almost all organ has circadian rhythms. And then manipulation of such kind of diseases or drug, which is usually used for the treatment of such kind of diseases, are circadian dependent. And then now we have a kind of a tools like a wearable to track the circadian rhythms or maybe method to measure the single time point, or how to say, based on the single time point measurement, you can track the internal time. So by using that kind of tools, we may have a kind of a leverage, even though we are a very small number of people, but we can, how to say, have good influence on the many different type of the diseases. I believe so. Yeah. But I guess at the same time, for young people who are looking for a new career,
Yeah.
this is very fulfilling
Mm-hmm.
because, as you pointed out, you started from researching circadian rhythm and computer modeling them,
Yeah.
and then going to study them in fruit flies,
Uh-huh.
to now you're talking about taking a leadership role to convince the Japanese healthcare system to implement sleep
Yeah.
monitoring
Yeah.
as an annual health checkup. So I don't think there is any other profession, any other branch of biomedical research where you can go all the way from modeling and
The society.
to biochemistry to imaging to personal health, public health, and societal
Right.
health. Right.
Right.
So
Yeah. I think, yeah, so you're right. So because sleep and also circadian rhythm are related to time, and then time usually go beyond from the spatial layers, like atom or to society, right? So that's a unique aspect. So and also it's fascinating, and you can go back and forth between different layers, and then and also that's natural in biology, because when some event in your brain, maybe some event in your neuron or protein or channels can influence your entire behavior, right? So therefore, space and time, or how to say, are connected with each other. And then some small molecular or atomic event can influence to the organism or even societies dynamics. So I'm always fascinated by the kind of a biological timing. I'm sorry, I sell my own topic, but I think that's the beauty of the psychedelic research and also sleep research.
Yeah.
So I hope current graduate student or undergraduate student are fascinated by the such kind of nature of psychedelicism as well as sleep. Yeah, and then jump into the uh, new topic in terms of the you know rhythm or sleep.
yeah. Well, I guess you also brought up like the physicists, mathematicians, technologists, and engineers. Um, there is space for everybody.
Right.
Yeah. So I guess with that, I would like to thank you again because this has been really exciting to go through a whole breadth of science from models to society. And you have been really transforming this field, pushing the limit. And I hope.
So we, right?
Well, it takes a village to move science, so but we all have our unique contribution.
Right.
And thank you so much, Hiroki. And
Thank
hopefully,
you, Sachin.
we'll
Yeah.
see you again
Yes.
soon. And hopefully, I'll get to talk to you again in next year or so.
Yeah.
There'll be many more exciting stuff.
Yeah.
Thank you.
Thank you.
I'm your host, Professor Satchin Panda. I hope you learned something new and found this episode exciting. If you did so, please leave us a comment. If you like the science that we discussed and would like to make a donation to research in my lab at the Salk Institute, you can do so by going to panda.salk.edu/giving. Thank you again for listening, and I'll see you next time.